So you might have to cut out
people
some
did
stuff.
that. It's highly editable. We're not live, so oh shoot, I don't need to bump into that. This thing, I have a love hate with his cameras, Chad's, because he's got this. It's like a gimbal, it just goes any direction. But it's once you let it go, this camera's so heavy, it just.
Oh.
So I try not to.
Tighten it.
Try not to mess around with it too much because I've got it the way I want it. All right, so your paper is in the shot though, so let me move that over a little bit.
Is my water?
Your cup is not.
Okay.
No. It's just kind of like about a foot of that side. So.
Is my lap in the shot?
Your lap is not in the shot, so you could keep that there as long as we get your hand gestures. Make sure everything looks good.
I'll just put it.
And if you need to take a break and look, that's totally fine. Or if you're like, what was I gonna say? That happens. It's also fine because it's all very editable. The whole idea is I want you to be as comfortable as possible.
Yeah.
We're just having a conversation.
Yeah.
So you look at me. These do not exist. They're not here.
Yeah. I was like, didn't want it to be like rehearsed and it's supposed to be an organic conversation.
Yes.
So.
I want because otherwise things that are so rehearsed, okay, here's your lines, and and it to me that doesn't always come off very well.
Yeah.
It's what people want to know about when they work with us is, okay, who are they really though? Mm-hmm. It's almost like dating in a way. It's like, okay, who we're going to be partnering with these people for a long time.
Yeah.
We want to make sure they're nice to work with, kind of thing. And so that's kind of the idea. Is like these are the people behind what we do and why you'd want to work with us because we have great people. Mm-hmm. That's pretty much it.
Okay.
Good.
Mm-hmm.
Cool. All right, I'm going to take a drink of water. And I will do a brief intro, and I will double check the pronunciation. Is it Rasmussen?
Rasmussen.
Okay, I was way off.
That's okay.
Ras or Raz?
Rasmussen. Yeah.
Rasmussen. And the emphasis on the Ras.
Yeah.
Rasmussen. Megan Rasmussen. Way better than I was trying to say it.
Rasmussen is a very East Coast pronunciation, I think. Some people do pronounce it that way.
All right. Rasmussen. Mm-hmm. All right. Deep breath.
Me too.
Yeah. So today I am sitting down with Megan Rasmussen, a senior scientist in molecular and cell biology at Solarion. Megan works at the intersection of complex cell-based assays and real clinical samples, and so I wanted to talk with her about what she thinks about the impact, the problem-solving and responsibility in this kind of work. Megan, what's important to you in life that directly or indirectly kind of affects what you do at work?
Yeah, so I really enjoy helping people, and that's always kind of been my main goal in my career is to do something that would go on to help people. And so being able to do that through a research-based position has been really rewarding, and so that definitely relates to my day-to-day job, being here at Solarion and being able to contribute to drugs moving through clinical trials and ultimately getting to patients.
It's fun, isn't it? You get to marry science and the enjoyment of everything that you've learned through your graduate program and actually apply it to actually help people. Mm-hmm. I think it's why we love being in this field. Now, you've worked across academia, pharma, and now CRO environments. What changed for you as you moved closer to clinical development?
Yeah, so originally I had always wanted to be close to the clinic, and doing that through kind of a more basic science approach was really what I ended up doing. And so really my graduate work was very basic cell biology mechanisms, including cell death, and that kind of lent itself to looking at anti-cancer drugs during my PhD. And knowing I wanted to go more the industry route, I ended up moving on to pharma for my postdoc and first position as a scientist. And that happened during the COVID pandemic, and so we were working directly with mRNA vaccines and kind of developing the next generation of those types of vaccines. So it was really fun to be able to be still in a preclinical environment where you can be very creative and open to learning new things and using different techniques, but being able to apply that to an actual, mRNA vaccine backbone was really fun. And then moving to CRO, I feel like that part was the most surprising for me, was being able to literally be right above the clinic, which is where I wanted to be, being near patients and clinical trials, but being able to help our sponsors get their drugs to patients by developing assays and being able to use all that past experience in developing the most robust assays we can, and still being able to still use creativity and innovation in developing those assays to get the best data we can.
Awesome. What surprised you the most shifting from basic research to applied to clinical work?
I think, yeah, I kind of maybe had touched on that a little bit in my last answer.
It's okay. We can
So yeah, kind of being able to still use like creative thinking. I think people think that going to a CRO environment, you're just going to do the same thing every day, maybe, and that's really not the case at all. Like every project is different. Every project comes with its own challenges, and it's really fun to be able to solve those challenges and then still be able to see a clinical trial go from beginning to end, especially in my position in the molecular and cell biology group. Like we work with the sponsors from the very beginning of the assay all through the production aspect where it's actually being used in the clinical trial, and being able to see that big data set at the end where you can see that their drug is making it to the target and having the effect that they want to see. So that part, I guess the satisfaction that comes from that has been surprising in a really good way.
That's awesome. Yeah, no, and it is. You went into the mRNA vaccines, and that was a crazy, exciting, huge problem to solve situation for so many scientists that were rushing to get something across the finish line that could help people during a pandemic, which we didn't think we would have to live through, but we did. Mm-hmm. And it was impressive how scientists were able to get to the finish line faster than we thought before, because the problem drove the need for the solution. Mm-hmm. It's cool. So for someone outside the biotech arena, how would you explain what you work on and why it matters?
Yeah. Let me look at my notes really quick.
Yeah, take your time. Whatever feels right, and if it comes down to it, we don't even have to go there. It's not, really what I'm trying to get out of this, to be honest, are 2 or 3, really great, impactful, meaningful, organic moments that people are like, wow, that's really cool. Yeah. And that would inspire people, whether they're a sponsor or a scientist or just someone who, thinks medical innovation is cool. Mm-hmm. So do you want to skip that?
We can talk about it. Okay.
Yeah. So I'll start over. Okay. So for someone outside biotech, how would you explain kind of what you work on and why it matters in the world?
So specifically in our group in molecular and cell biology, a lot of what we want to understand when a drug goes through a clinical trial is what is the pharmacodynamic effect, or what is the drug effect on your body, and is it making it to the target that we expect it to go to? And that can be really hard to see in healthy volunteers that don't actually get sick, and so you can't actually cure them of anything. So we get to really delve into the mechanism of how the drug works.
Yeah, no, and that's great and important, and I think a lot of people don't realize all the different steps and how long it is to get from an idea or a molecule to the market with an actual medicine. That's cool. You work on a lot of, your work involves a lot of assays that are technically sensitive, or maybe even hard to stabilize. How do you usually approach problems when things don't behave as you would hope they would?
Yeah, so personally, I go to the rest of my team. We all have a lot of different expertise. We've all come from a lot of different backgrounds. So normally we'll all kind of put our heads together and think about ways that we could stabilize. The stabilization, for example, is a huge topic in the field. And so oftentimes going to like the AAPS forums online is super helpful. Going to those folks that have published those papers, looking through the literature, seeing what can be done and what we can implement here in our CRO environment where things need to be really robust, really scalable, and transferable to our analyst pool who is actually handling samples every day.
That's great. And I think it's something that is remarkable about our industry is that there are people who may even be competitors that are willing to share information about how we can get to those endpoints faster, more efficiently, better quality, more efficacy, and so forth. And that's really what drew me in, because there's actually this collaboration. It's almost, you go to these conferences and it feels like it's a commercial level conference, but it feels like an academic conference because the way people are relating to each other.
Yeah.
And I think that's outstanding.
Yeah. Kind of related to that, like there's a PBMC working group that's been started by folks from other companies, and it's across CROs and across academia, and everyone's really trying to put their heads together specifically for PBMC isolation, stabilization, best practices. So that's been really fun to be able to be a part of that from people all over the world, and just trying to put our heads together to come up with more standardized ways of doing things, especially in like the biomarker field, because it is so different with every assay and newer to the regulated world, I would say.
I think it's really great and essential to what we do in getting success faster. Excellent. What does good collaboration look like to you when you're working with teammates or supporting a study team?
I think ultimately coming into things with a positive attitude is the biggest helper with making collaborations go smoothly, whether that's, we're in the lab trying to solve a problem, or, collaborating with sponsors, because really they are collaborators, not really, if the relationship is working well, it feels a lot more like a collaboration, which is a really fun part of what we do here at Solarion. And then again, I mentioned like collaborating with the rest of the people in my team and bringing in lots of different people with different knowledge and different expertise, and going to the people who have been in this industry for a long time. They're often, they have a wealth of knowledge that's always super helpful for solving different problems. So.
Excellent, excellent. And as someone who mentors and works closely with others, what kind of environment do you try to create on your team?
Yeah, I always just try to make things really positive, and when I'm excited about an assay, I feel like that can translate over to other people who may not have the depth of knowledge that I do about the assay since I developed it, but I try to get them to that level as they learn how to run the assay, especially with these more complicated flow cytometry assays that we're doing, or ex vivo stimulation. A lot of them have really great questions on, why do we do this step, or what is this drug actually doing? And I love when people ask me those questions, because then we get to delve deeper into it, and then they kind of take over ownership of that assay as they run it every day, and they really get very good at understanding what they're looking at and what makes a sample look good on the flow cytometer. And I love when they also follow up with me like, hey, how's the data been looking for this assay because we're doing things in real time, so it's really fun to be able to kind of give that feedback back to the team. And I feel like that helps them, stay motivated and know that they're doing a good job, and ultimately that will just lead to better data, so.
Excellent, and I'm sure it does.
Mm-hmm.
So speaking of motivation, what keeps you excited or curious about the work you do day to day?
Yeah, I guess, what am I going to say? Let me look at my notes
Yeah,
again.
take your time.
I feel like I just end up repeating myself for different things.
Well, I've kind of, I feel bad because you're answering these questions so well that you're kind of answering, sometimes two at once. So, and we can always, consolidate some stuff so it's not repetitive.
Oh yeah, the last question that you said. Okay, can you repeat the question?
Yeah, no problem. Start over. So Megan, what keeps you excited or curious about the work that you're doing day to day?
Yeah, so I really enjoy the opportunity to be able to work on many different drugs and many different platforms, and also I've learned so many different techniques since coming to Solarion, so I really feel like I've been able to expand my breadth of knowledge both technically and also kind of about regulated clinical trials. I've never worked in that space before, and also being able to work with lots of different sponsors who all have different drugs and different mechanisms of their drugs that they're looking at. So really just being able to have a wide variety of things that I'm working on, like no two projects are the same, so that really keeps me interested and excited to learn more.
I agree. I think that's the fun part of the CRO is that you're not stuck to one compound for a very long time.
Mm-hmm.
You get to explore different molecules, different technologies, and different indications on a regular basis.
Mm-hmm.
And you get to see those, even if you only touch a piece of that, for a little while at a time, you can look back if it makes it to market and say, ooh, I worked on that drug.
Mm-hmm.
I developed an assay for that. And
Yeah.
it feels nice to be a part of, improving healthcare.
Yeah, that's definitely one big thing from academia and even pharma, or at least my pharma experience was that you have one project and you might have different aspects of that project that you get to work on, but you're kind of like, that's the goal, and the goalposts are always moving. And in this job, it's much more fast paced. You have milestones that you want to hit at certain times, especially with our assays that need to be run fresh. So it's like as soon as we start talking to the sponsor, we have a timeline that we need to hit. So it can be high pressure sometimes, but I feel like that also keeps things interesting.
Yeah. Well, as Tyler put it earlier today, the pressure is a privilege,
Mm-hmm.
which means we're working on something important.
Yeah,
Because it's hard.
absolutely.
I thought that was pretty cool.
He has told me that before.
Yep. Yeah, I think that's one of his axioms.
He's full of good things, good
Yes.
little quips.
Well, Megan, thank you so much for spending some time with me to share how you think about the science and the responsibility behind it. I appreciate the care and perspective and the positive energy that you bring to your work and to everyone who works here.
Thank you.
Absolutely.
Thanks for having me.
Cool.
All right.
That was fantastic. Exactly what we wanted to get.
Cool.
Yeah, this is perfect.
I'm excited to see. It'll be like, will it be